And we'll go through the different ways we classify them. You guys already know everything about this because you've been in the lab, right? So we're going to have you guys teach me. All right? What's that? No, I'm just saying, since you guys already know all this, I'm just going to ask questions. Okay. All right, so our two divisions of prokaryotes are archaea bacteria, or sometimes just referred to as archaea, and eubacteria, or sometimes just referred to as bacteria, okay? So it can get kind of confusing because archaea sometimes are included within the bacterial group when you're talking about single-celled prokaryotes. But technically they are not included. But they do have some specific differences, including the bonds in their lipids. Okay? So from the video lecture, there were two types of bonds. What were the two types of bonds in which ester and ether, and which one went where? So was ester with archaea? Bacteria, right? So ester bonds and ether bonds in the lipids. There you go. That's why you take them. All right, and so there's also an important structure which is made of sugar and protein. Okay, and what am I talking about? Peptidoglycan, which means, what's a peptide?
Protein.
All right, yeah, it's a protein. What's a glycan? What does that refer to?
Sugar.
All right, so it's a protein sugar. Okay, so which one has peptidoglycan? Bacteria, yes.
Would you see peptide, can't pronounce it,
Peptido.
in like fruits? Is that where you see that source? Or like
No, it's only in bacteria,
bacteria?
as far as I know, yeah. So archaea does not have peptidoglycan. Okay, and peptidoglycan, the amount of peptidoglycan determines whether it's gram positive or gram negative. Okay, and then there were a couple other things which also helps us with our universal phylogeny. All right, so you remember this? Our three different domains. Okay, which one is over here on the far left? Good, bacteria. And then we've got archaea and eukarya. So, bacteria has a transcription and translation process that is unique to the bacteria. Okay, whereas archaea the process is similar to eukaryotes. Okay, so where would you, if we were going to write the character for transcription and translation, where would you draw it in our universal phylogeny? All right, so we draw it right down here, right? The process. So this indicates a common ancestor between those two. Not the both of them go through transcription and translation, but the process is unique. Okay, and bacteria also have a unique replication. Where, yes?
Sorry, you were probably about to explain replication a bit more. I was just a little confused, even after watching the lecture, what was unique about his replication.
So,
Transcription and translation.
yeah, what was in the video lecture? What about the replication was unique? Do you remember? Yes, single origin of replication. Okay, so replication starts at one point, and then it goes in both directions till it completes the one chromosome. Okay, where in archaea, replication is similar again. To eukaryotes with multiple sites of origin, which would also allow it to, which works better for a longer, more complex chromosomes, which eukaryotes have. Okay. All right, so those are some major differences between the two. Archaea, we also have archaea are extremophiles. And are much more rare than our bacteria, which you can find pretty much everywhere. Okay, so, and beyond kind of this very specific instance where we're talking about these differences, any other time when we refer to bacteria, we're just referring to this group. Unless archaea is specifically mentioned, then bacteria is assumed. Okay, so then with bacteria, we can classify them based on different characters. So I'm going to draw some shapes, and you're going to tell me what this is, right? What is the term for this shape? Okay, coccus. Yeah, it's not a circle, that's a sphere. See, I need my chiaroscuro shading in here. Okay, see that? All right. Rod-shaped bacillus. Okay. Spirilla is what I was trying to draw. Okay, and then there's one other one. The comma-shaped.
Vibrio?
There you go. All right, and then we have aggregations of those. So, and we'll just talk about two of the main ones. So staph, what does that mean? It's not a chain, it's a, yeah, cluster or a group, yeah. And strep then is a chain. Okay, so a streptococcus looks like what? Yeah. There's streptococcus. Staphobacillus would look like what? Yeah, so all these guys would be stuck together. Okay, good. And you guys know this stuff. It's back of your hands, right? These other structures, then, accessory structures, are not necessarily found on all prokaryotes, but are important if they have them to understand. It helps to classify them. So our prokaryote, and I'm going to attempt to draw them on this little prokaryote over here. So a prokaryote has a cell wall, right? And let's say this cell wall, this part of the cell wall in red is the peptidoglycan layer. Okay, it may have another layer outside of that. Steady hand. Called the S layer. And it can be used for different things, but mostly is used for aggregation, so getting together and sticking together. All right, it may have, if it has one, it's going to appear outside the peptidoglycan layer. In addition to that, it may have another layer. I'm just going to draw it on this side. A capsule, which is a gelatinous layer. Which adds another layer of protection to it. Some of these pili, flagella, and cilia look similar, but have different functions. So flagella are generally really long, okay? And they may have multiple flagella coming out of different points. So they may even have flagella on both sides. But the flagella is for locomotion. Moving around. Cilia are shorter and more numerous, but it's also for locomotion. But the way that they work is different. So whereas the flagella just kind of whips like a motor in like a circular pattern, the cilia basically move in waves, and those waves then help propel it. All right, pili then are longer than, I'll do a different color. Are longer than cilia, usually shorter than flagella. And these are not for locomotion. So these are actually for attaching to other bacteria and pulling them closer. So also for aggregating. Some bacteria are able to create a dormant state. Okay, where the cell itself would actually shrink to this very small, or at least much smaller than its normal size, which with a very thickened cell wall. And all of the cytoplasm would be reduced, and it would form then this endospore, which is better able to withstand conditions which aren't ideal for its growth. So if it's too cold or too hot. Yes?
So the endospore itself is the bacteria that you just drew, but just in its dormant state. So the flagella, they're not there. My And it's dormant state. Just,
Right.
okay.
Yeah. So it's just going to have the basics it needs to proliferate again once conditions are right.
So the endospore is inside all the other layers. It's not on the outside of the.
No, basically this cell shrunk and formed an endospore. Yeah, and all those. Well, yeast is a eukaryote, and it's not as hardy as an endospore. So botulism, the bacteria that form botulism make these endospores. And botulism is one of the reasons why we pasteurize all our canned goods. You have to keep it at a certain temperature for a certain amount of time, and that will kill the endospores for botulism. But if you don't, then they may survive. All right, and then they may also have various complex membranes. Okay, so this plasma membrane may fold over inside of itself and create a photosynthetic membrane. So in a cyanobacteria, it's going to have a structure similar to a chloroplast. Or if it can go through cellular respiration, then it may have a membrane similar to a mitochondria. And these can often take up most of the cell and allows that, that cell to participate in those very complex processes.
Is it complex?
Complex membrane, yeah. So this would be a photosynthetic one, but it could also have other membranes for cellular respiration. Yes?
Do like the flagella like break off or get lost and like have to go back or anything like that?
That's a good question. I don't know. But yeah. I imagine they're pretty integrated into the cell wall, so if they break off, the whole cell would probably die. But I don't know. Okay, so those are the different ways with these cell complex membranes. Bacteria can go through aerobic respiration, just like eukaryotes can. Just specific ones, not all of them. Most of them cannot. Some bacteria can go through photosynthesis. Right? Cyanobacteria is a very large group that can do this. Classified bacteria. All right, here's some questions for thought, and we've kind of talked about this before. So again, mention. Okay, so this is oxygen levels, which have changed over the course of our Earth's history. And what, just for review, what's the equation for photosynthesis? Okay, see if you can remember with your neighbor. Okay. All right, what's on the left side for photosynthesis? Okay, we got carbon dioxide and water. Thank you, Google. And on the right side, you then have okay, and if you wanted to balance them, yeah, you could put this. All right, so then what's the equation for cellular respiration?
Pretty much the thing backwards.
Yes. C6H12O6, that's our sugar.
Take a sugar and an oxygen and it creates CO2 and
Okay, so knowing that, where do you think the following four mechanisms of cellular function occurred when looking at the timeline? Discuss with your neighbor, then we'll talk about it together. I numbered these just so I could put them on there easier. But let's start with number 3. So let's start with number 2, anaerobic bacteria What does anaerobic mean? Without oxygen, yeah. Anaerobic bacteria generally die in the presence of oxygen. Okay, so they can't survive in a high oxygen environment. So where in this timeline would you have organisms dominated by anaerobic bacteria? Before oxygen, right? So this whole area here is going to be when most of the species which covered the earth, most of the bacteria, were anaerobic. Okay, but what occurred then at this point which caused then oxygen to increase in the environment?
Explosion of anaerobic respiration.
Anaerobic respiration?
No, sorry, photosynthesis.
Yes, photosynthesis, right? Because we said a byproduct of photosynthesis is oxygen, and that's what caused then an increase in oxygen in the atmosphere, right? So you probably have that occurring at this step, photosynthesis. Okay? Once this oxygen then starts to increase in the environment, now all these anaerobic bacteria are going to die. Right? And after then oxygen is available, then you can have aerobic respiration, because we can't do aerobic respiration without oxygen. So probably sometime after this, once you have enough oxygen in the environment, you can have aerobic respiration. All right, when do you think anaerobic respiration then occurred? Where did you guys put that?
Comets hit the earth.
Okay. Well, you would need water for it.
Bacteria, too. They're anaerobic bacteria, and they have anaerobic respiration, right?
So anaerobic,
Or is that what you're talking about?
no, cellular respiration, but without the use of oxygen. So that would be just somewhere on this side, okay? So using a mitochondria-like process, but you don't need oxygen to do that. Okay, so some extremophiles can do that. Archaeans. All right, but yeah, so probably evolved before aerobic respiration. Aerobic respiration then evolved after you had an increase in oxygen. Something like that. Okay, does that make sense? Any questions? Okay. All right, so we did a gram stain in lab. These are the 4 different things that you put on your slide after you had your bacteria on there. In what order did you put them on? I just mixed them up. So if you were to do 1, 2, 3, and 4, 1 being first, can you remember with your friend which order they go in?